And then we also have students from environmental engineering, I think. And we also have some audience that we invited for this occasion. And today I will be the moderator since Pak Tjandra has another engagement in this morning. So I would like to welcome Prof. Raj to give the lecture.
Thank you very much, Arjun. Good morning, everyone. It's good to be seeing you again one more time. This lecture is going to be, as Dr. Arjun said, this is biological treatment of wastewater. It's because of the mixed group of audience. I kind of start with basic information, and then I'm going to talk about some of the biological reactor, and then what is currently practiced in the US, and then I will finish with case studies. So some of you might already know what I'm talking about. So you have to bear with me because of the mixed group of audience, okay? Let me start with my first slide. So first of all, if you look at the wastewater we generate worldwide, only 10% of the wastewater is being treated. 90% of the world wastewater does not receive any treatment at all. So it just goes straight into the moving water bodies. So that's a fact, okay? Only 10% of the wastewater worldwide produced being treated, okay? So what is the concept of wastewater treatment? Basically, we generate the carbon, organic carbon in the wastewater. We want to reduce them. If there is rich in organic carbon that goes into the water body, what's happening is the aerobic bacteria are going to consume the carbon, and as a result, it's going to decrease the dissolved oxygen in the water. As a result, you're going to see fish kills. A lot of fish all of a sudden you see dying in the rivers. So the concept is you wanted to reduce this carbon load into the wastewater. By encouraging the growth of microbes, the one limiting factor is the oxygen. So you provide oxygen. So when you do that, microorganisms are going to convert this carbon into carbon dioxide, and they're going to produce more biomass, going to produce more salt, and then water is produced during the biochemical reaction. The limiting factor is oxygen. So most of the time you aerate the wastewater and allow the microbes to grow on the carbon. And then, of course, there are wastewater that contains other material like nitrogen, phosphorus, and you also have some hazardous chemicals. So you need to make sure what type of wastewater you have, characterize the wastewater, and then sometimes you have to introduce special microorganisms to do that. So the general concept is to reduce carbon loading in the wastewater before treatment, and when it is treated, it should be within the acceptable limit of the regulatory agencies in every country. So since it's a mixed group of audience, I'm going to start with the definition of few terms, because most of these wastewaters, you start with the loading based on the BOD, COD, and solids. So just a fundamental definitions. So BOD is defined as biological or biochemical oxygen demand. It basically defined as the amount of dissolved oxygen needed by aerobic bacteria to completely break down organic materials in a given volume of water at a certain temperature over a specific period of time. Usually, they incubate BOD 5 days, and they call it BOD 5, sometimes BOD 3-day incubation at 20-degree temperature to analyze how much carbon is in the water. So BOD is basically what they're measuring is the biologically degradable organic matter in water. So if there is more carbon in the water during the incubation time with this sample, the more oxygen will be consumed. When that consumed, there is a relationship between the carbon concentration and oxygen consumption. Another term is COD, which is chemical oxygen demand. It is the measurement of chemically oxidizable carbon in the water, organic carbon. So this includes both biodegradable and non-biodegradable carbon. There are two kinds of COD. We measure total COD, which is taking sample and chemically digest the sample and look for how much carbon in the water. That include the contaminant as well as the microorganism, because microbes are basically containing 50% carbon. And then the soluble COD refers to only, you filter the sample. So what is dissolved in the water is what you're measuring. So we have total COD and soluble COD, which exclude biomass in the water sample. The next definition is a lot of solids also. When you talk about wastewater, you need to know how much solids go in there. So we have total solid, suspended solid, dissolved solid. So the regulatory agencies make standard based on how much solids in the water, because you want the discharge water should be as clear as possible. You don't want to have turbid water going in there. When the water is turbid and the receiving water body gets turbid, and then that's going to affect the light going through, that's going to affect photosynthesis. So you need to make sure your solid is less when it is treated and let it go. So total solid is basically all particulate matter that remains after evaporation at 105 degrees as a residue. Suspended solids are the particulate matter that are suspended in water, so that you filter the sample and how much a sample is filtered, the solid that's filtered is suspended in the water, right? That is called suspended solid. If you know this total solid and suspended solids, the difference is dissolved solid. The dissolved solid is mostly the ions in the water sample, okay? And you already know what is a reactor. Reactor is a container, a vessel in which you make the biological reactions carried out. And then the organic loading is basically the amount of carbon that is loaded into the reactor when you treat the wastewater. So basic features of wastewater system is you should have primary, secondary, and tertiary treatment. Primary is basically physical process. You screen the wastewater to remove any floating material, and also allow the wastewater in a sedimentation tank to remove solids. So basically remove any physical material in the wastewater. Secondary treatment is where the biological process takes place, and this is where you allow the bacteria to multiply and provide oxygen, so they reduce the carbon in the water. So this is where the biological process takes place, secondary. And tertiary is mostly chemical process to kind of remove the pathogen in the water. So mostly chlorination, ozonation, precipitation, adsorption takes place in tertiary treatment. So when you design the wastewater system, you need to take into a lot of things into consideration. You should know wastewater characteristics and how much volume of wastewater is generated in a site. So you need to design based on the volume generated. So you need to know how much loading rate goes into the system, how much flow rate going into the system. Okay, so the basic concept of designing wastewater is based on this. The HRT and SRT. Hydraulic retention time is basically the time the wastewater spends inside the reactor, okay? Solid retention time or sludge retention time is the amount of time the bacteria, the biomass, is spent in the reactor. So when you design a reactor, you need to have high SRT. You want to keep your bacteria in the system as long as possible, because these are the bacteria that is removing the carbon. You don't want to lose this wastewater along with a treated water. So you need to try to retain this biological solids as much as possible. But at the same time, you need to treat your wastewater as fast as possible, because time is money. You want to treat them as fast as possible to meet the regulatory standard. So you want to keep your HRT short, your SRT long. So when you design a system, that's what most of the engineers calculate and do this process. And then you do process analysis. You need to basically analyze what is going in, which is called influent to the reactor, and the COD in solids and and BOD. And then after a certain amount of HRT and SRT, what is coming out, and you analyze that. And then you do the performance of the reactor. So how much COD is removed, how much solid is removed in the system. And then you need to do mass balance. So you how much is removed, how much is still inside the reactor. You calculate and come up with the carbon mass balance or biomass mass balance. You can calculate that. So having said that, basic parameters, if you look at all the commercial reactor, they are based on 4 principal type of reactor. There's only 4 principal type, but all the commercial reactor follow any one of these 4 principal type. The first one is batch reactor, which is it's a batch system. There is no influent, there is no effluent. So you fill the wastewater in a tank and allow the reaction to occur. And when the COD, BOD level goes down to the regulatory standard, you pump it out and wait for the next batch to come, okay? The liquid, of course, there's a mixing, so to get a better mass transfer. And the next reactor is called plug flow. Plug flow is also sometimes referred as tubular flow reactor. Here, the particles that enter the tank are discharged the same sequence in which they enter. So there is a diffuser put in the influent point, so they go through the same level it comes into the system, it goes out. So basically, there is no mixing. So you save some money for not putting mixing device. The particles retain their identity and remain in the tank at an equal time or theoretical value that you set in when you design the reactor. And then CSTR is called continuous tank reactor. This is where complete mixing takes place. And the particles are completely dispersed, so you have better mass transfer between bacteria and the biome and the carbon in the system. And it is completely mixed, and there is an influent, there is an effluent, so it's a continuous system. The wastewater coming in, the wastewater going out. So this is where you lose your bacteria when. So you want to keep this SRT long. You want to try to keep the sludge inside as long as possible. And then the arbitrary flow is a kind of a partial mixing in between a plug flow and a complete mixing. So you put a baffle inside the system. So instead of putting a mixing device, the baffle will create a partial mixing in the reactor. So any commercial reactor falls within these 4 principal type of reactor, biological reactor. So let's look at some of the pictures. A batch reactor is simply a batch system which have a mixing device, and initially you load them and then mix them and allow the whatever HRT is, you set it up for the BOD, COD to go down once it's reached it, and then you discharge the wastewater and wait for another batch to come in. CSTR is almost it's like a batch, but it's a mixing, but it has an influent and effluent. Wastewater coming in, wastewater going out in the system, okay? It's a continuous process. And plug flow is like a linear flow, and it's also called tubular flow reactor. The wastewater that comes in is a diffuser. Each one goes in the same direction it entered, so you don't see a mixing. It just goes. So it's just throughout the reactor, the wastewater is distributed. So you don't need mixing, right? So if you look at another way of thinking about this resident time inside the reactor is that actually it's basically the volume and flow rate you set, then that will calculate the resident time. Okay, the time spent in the reactor is equal to all elements. So there is no mixing, it just comes and goes. During that process, the microorganisms interact and remove the carbon in the wastewater. That's another design to look like the plug flow reactor. So you have a continuous process, waste goes in a tubular form and then comes out. During this HRT you set in, bacteria will work on the wastewater to remove the carbon. And this is called the, they put a baffle in there, so you have a partial mixing, we call arbitrary flow. So here you don't have to have a mixing device. So the wastewater coming and hitting this baffle, and it creates a partial mixing without using energy to mix it. And we call it arbitrary flow reactor. So now I'm going to show you some of those reactors. So all of them follow one of these principal type of reactor. Yes, here. So when you design a reactor, you need to know what process you're applying for. There are a lot of wastewater out there, right? From not only in municipal wastewater, but different factory producing different chemicals, food industry, leather industry, and all kinds of industry. You need to look at what kind of waste you are dealing with. And again, you need to know the volume of waste generated. You need to do the analysis of wastewater, and then look at, is there any toxic chemical in there that can inhibit microorganism? If that is the case, you need to design a pretreatment. And then, of course, in Indonesia, it may not be applicable, but other places, there is a temperature issue. If it is like cold countries, cold zone, you have, during winter month, it's going to have a performance problem if the temperature is too cold. And then climate, and then we have to also do the reaction kinetics, what's going in, what's coming out, what rate it is being removed, make the calculation, and do the performance analysis of the reactor. You also need to know bacteria, there is some nutrients that is missing, you may have to provide nutrient to make the bacteria happy. And then, of course, you need to calculate how much energy you're going to use to operate the reactor. And then the reliability and complexity of the system. So you want to keep this as simple as possible. You don't want to make your reactor too complex, okay? And make sure it is compatible with, if there is an existing system there, try to make it compatible. Don't create something totally new, okay? So when you get that, set up the reactor, then you follow this characterization. You need to select particular reactor, and of course you need to do treatability study. If you're asked to design a system, you need to do in small scale in a lab, make sure everything is working, and then you do a little scale up and do a pilot plant testing, and then you do commercial reactor design. So this is the way everybody operates. Then you need to make sure you leave options for modification, because once you set it, sometimes the factory modified systems, they don't produce as much wastewater. Sometimes they produce more than you designed for. So you need to make room for a modification if necessary when you design a system. So some of the commercial reactor, let's look at some common one. This is the Aflo anaerobic sludge blanket reactor. So here, what we are trying to do, as I said, you want to keep your biomass inside as long as possible. So what they create here, they create the bacteria in the sludge bed in the form of a granule. So the granules have a high settling velocity. So they settle at the bottom of the reactor. So when you pump in this wastewater into the system, so you don't lose this bacteria in the granule form, you keep it. So, and then the effluent goes out. This is a continuous system. Of course, this is an anaerobic, so you collect the methane, the bacteria converted organic carbon to methane, so you can use this biogas as energy. So this is one of the most commonly used reactor in industrial settings. So here you have large volume inside the reactor, and you're not putting any filter, you're not putting any film. So that's going to have your volume problem. So you're actually keeping the bacteria in the form of granules in this sludge bed. That's one of the common commercially operated reactor. So another design of USB just to show you what's happening inside. So sometimes the granules move up and down, and it also create a partial mixing. So without putting any mixing device, you can have the gas it is produced also mix the gas bubbles, and the bed also moving up and down, going to create mixing for you. So you don't have any mixing device. Okay. Another type is fluidized bed reactor. In fluidized bed reactor, you have 1/3 or 1/5 of the reactor, you put fine sand. And so when you pump the wastewater, the sand moves up in the fluidized motion, and it creates mixing for you without any mixing inside the reactor. You don't have to put a mixing device. And of course, this in between the sand particles, bacteria grow and form biofilm. So when wastewater go through this system, bacteria is able to convert this carbon into carbon dioxide and completely remove COD and BOD. So this is another type of commercial reactor called fluidized bed reactor. And this is just to show you the fluidization process. So when the bed moves up, you can see the sand particles mixing, create its own fluidized motion. So you have this mixing occurs at the same time when bacteria remove the carbon. This is a lab setup of a fluidized bed, just to show how a lab reactor look like. So you can have this sand particle in there, and then you have influent effluent going through, and then you can look at the fluidization of the reactor. So sometimes you'll have clogging problem, but most of the time it works. Another type is fixed film. So you have this big reactor where you have a big film, mostly plastic sheets. You allow bacteria to grow on this film as a biofilm. So then you operate this waste reactor either upflow or downflow, depending on how you're operating. So when the wastewater goes through the system, it's going to pass through this biofilm and come in contact with the bacteria. And so it's all bacteria grown and attached. So you don't lose the biomass when the wastewater goes out of the reactor. So you retain your sludge inside. So you have no problem of washing out of bacteria. So that's another type of commercial reactor. But this is, all these reactors follow plug flow principle type, because the motion of the particles inside go similar to plug flow reactor. And then we have a early bio bed. He can put wastewater going through biological materials, and we call it bio bed. And then again, the same principle, bacteria is growing on this biofilm, and the wastewater is pumped through, the same concept. So you retain the biomass inside the reactor longer period of time. And then we have biofilter. There is a lot of filter material we can use, like ceramics, rocks, plastic material. And then inside this biofilter, we have again biofilm. You can see bacteria attached to this biofilm. And then the concept is again the same. You try to retain this biomass. These are the good bacteria. They are already remove the carbon. So you don't want to lose this bacteria along with the effluent. So you're keeping this bacteria in this biofilter, trapping them inside the reactor as long as possible. CSTR, so CSTR is sometimes really good, but it is energy intensive. You have to have this mixing device. You're spending some money to operate the reactor. And then the problem with CSTR, you're going to lose your biomass along with the effluent. So they modify the CSTR. They put settling tank, and to retain the sludge that goes out, allow the sludge to settle, and then you recycle the sludge back into the reactor. So you get this good bacteria back into the system. So the basic concept is the same. You want to keep your bacteria inside the system as long as possible. You don't want to lose it along with your effluent. And then you can have hybrid. You can combine, a couple of reactors together and create your own reactor depending on what type of wastewater you're treating. Here is your sludge blanket as well as filter put in there. You have this bacteria created in a granule form, and then you also put a filter on top. So you combine two systems together. We operate filter. Another common one is the Baffle Reactor. Baffle Reactor also follows the same principle of plug flow. So you put a down baffle and up baffle, and the wastewater follows certain paths. So wastewater comes in and then goes down and up and down and up through this baffle, and then comes out of the effluent. So you're keeping most of the biomass inside these chambers, so you're not losing the biomass out as fast as the water goes out. So there's another type of reactor called anaerobic baffle reactor, and then the methane is collected and used for energy purpose. Just to show you a lab setting of USB treating, it is a food industry wastewater. You can create your filter any type of design you want. You can have a horizontal flow, or vertical flow, or upflow, or downflow, depending on what type of waste you're dealing with. Okay. Another type of reactor is called RBC, which is rotating biological contact reactor. It's basically big biodisc, and these biodiscs move slowly. There's a motor in there, and your bacteria grow on this biodisc in biofilm. And every time they go down and touch the water and come in contact with the carbon and remove, and they go back up and get oxygenated, get oxygen. And you need to have a good viable biofilm in the system. Biofilm, there's a different degree of biofilm. You have anaerobic biofilm, aerobic biofilm in the same disc. So you have a lot of complex dynamic going on in the RBC. So this is another popular reactor out there in the industry. Just to show you actual picture of a biofilter rotating contact RBC, biological contact reactor. So you can see the motor here, the whole disc rotates, and there's a biofilm growing on this biodisc. Just to show you how these paddles are there, the disc arrangements are there. You can see it's whole pack. And then there's the shaft and there's a motor, and each one is attached to that, and then the whole drum rotates, so very slowly. And then we have the household type. If you live in a rural community in the US, you have the septic tank, and the septic tank could be treated in an anaerobic filter. You can, the same type of filter that you put different rock or ceramic or bead filters, and trying to go through the wastewater, go through this filter, and before you let it out in the moving body, because we have some strict regulations. So in US, what they do, the septic tank collects into the community, and they don't have a big wastewater. It's a rural community, and they'll have the anaerobic filter set up for, say, 100 households. These wastewater go in there, and then after they're treated, people monitor the COD in solids, and they let it out. Okay? And that's another design to show you how anaerobic filter look like. Basically, the filters are there to trap the biomass, and wastewater coming in, and the gas is collected. And sometimes you need to do nutrient addition like carbon-nitrogen ratio follow, but most of the time they work well. And just to show you what packing media is inside the filter. So nowadays there's a lot of plastic media, but when I started in grad school, it was basically big rock, and then they replaced with ceramic. Now we have this plastic media inside the filter. Just to show you how these different designs are located, you can have different layers of this biofilter, and you can, put a rotating arm and then move this wastewater distributed from the top, following the gravity instead of pumping it. Okay. Just to show you an actual working trickling filter system. So you have the filter in here, and the big rotating arm move and distribute the wastewater. So you have the wastewater going through the system the way it entered. And then it's go through the filter. The filter has a biofilm, and it's going to remove the carbon, nitrogen in the wastewater. Just to show you, sometimes you lose biomass. You can put a clarifier and allow the waste to settle. And sometimes you can recycle it. And then you can collect the sludge, and sludge goes separately. And just to show you this arm that rotates and trickles the wastewater down this filter in the trickling filter. Commonly, most of the municipal wastewater, the only thing they do is they aerate in a big pond or big tank. And that's called activated sludge, because the only thing that is lacking is oxygen. So the bacteria is there, carbon is there, enough nitrogen in the wastewater. You don't have to do anything except to oxygenate and keep it in a certain hydraulic retention time. And then most of the time you get 80 to 85% COD removal in this system. Okay. And then again, you can how to remove the solids from this aeration tank. You put a clarifier and allow the solid to settle down, and then the sludge goes to the anaerobic digester to treat further. And sometimes you have to recycle the sludge if you're losing most of the bacteria in your system. Sometimes you recycle the sludge back into the activated sludge process. You can calculate how much wastewater going in, how much is collected, and how much is retained in the form of sludge. You can come up with your own HIT, SRT in your system. It's just simple calculation of the volume of the reactor, how much time it spends. You can calculate how much sludge is produced, how much sludge is being recycled. Okay. So what is doing the job? Then mostly it's a heterogeneous population of organisms. Not only bacteria, we have fungi, we have yeast, we have algae, we have protozoa. There's a lot of organisms growing in this wastewater. You don't worry about keeping this as pure culture because you want to have as many bacteria or microorganisms in the system because your purpose is to remove carbon and nitrogen. So as long as they remove it, that's fine. But the one organism that do bulk of the removal in the sewage is called Zuglia ramagera, which is the organism we prefer. They remove bulk of the carbon in the system. And sometimes when there is a shock in the system because the wastewater is coming from different households, sometimes we put toxic material in the system, you're going to kill a bunch of these organisms. So in that case, there is a succession of microbes takes place. So you're going to wipe out the good ones because they are sensitive to this toxic chemical. And then you're going to have a filamentous bacterial growth, and we call that bulking. Okay, the bacteria that is responsible is called Spiritualus natans. It's a filamentous bacteria. It started to float, and instead of settling in the waste, the whole waste going to float in your wastewater. So we have some problem to address how to control this bacterial system in the flock. Just to show you another picture of different filamentous bacteria in the system. And you have all kinds of organisms. You have yeast, you have protozoa, and rotifers, and a lot of different organisms growing in this biological system. And then you can get as sophisticated as you want. Here you can, this cryogenic plant, they're using pure oxygen to, pump into the wastewater. They're spending money. Sometime the wastewater process get more sophisticated. Yeah, so pure oxygen is pumped in this one. And then just to show you, another big system showing the settling tank, a big clarifier. So what other design parameters you need to know? You need to know the volumetric loading, how much BOD or COD that going into the system, and then the food to microorganism ratio, what is the biomass, what is the carbon in the system. So we need to calculate that because you don't want to starve the microbe, putting less carbon, or you have more microbe and the other way around. So you need to make this in balance. And then when you're recirculating, you can have the recirculation ratio. So you need to consider these into account when you design a bioreactor. So far we talked about carbon removal, but nitrogen is another problem. So when you have nitrogen in the wastewater, if it is not removed, then you're going to have eutrophication when it is let out. So here is the beautiful North Sea in Norway. Norway is supposed to be a pristine country, but they have frequent eutrophication because of nitrogen in the ocean discharge. And you can see in the stream in Norway, the algal growth. That's at mountain elevation at 1,200 meters, and elevation was high, but even there, the freshwater has a lot of algal growth because of nitrogen in the discharge in the wastewater. You can see the algae growth growing in this pristine, supposed to be a pristine environment. And the other extreme, if you don't treat the wastewater in poor countries like Nepal, you're going to have less dissolved oxygen, high BOD in the wastewater. So you hardly have any treatment going on, wastewater going right into the river. And this is where you're going to have a problem. You can see big foam coming up. Basically, there is no oxygen, 1 milligram per liter here, and then high BOD in the wastewater. You're going to see fish kill and other, actually we call it anaerobic septic system taking place. So in order to remove nitrogen, you follow nitrogen cycle. Nitrogen cycle is, the ammonia is converted to nitrite, and nitrate by these two organisms encourage the growth of nitrosomonas nitrobacter they oxidize ammonia into nitrite and nitrate. Nitrous ammonia, nitrobacter will do this job. And then you turn the system into anaerobic mode, and denitrification will take over and convert the nitrate back to the nitrogen gas into the atmosphere. And just to show, you can do this in one reactor in a sequential operation we call the sequencing batch reactor. Operate first aerobically and then anaerobically and remove the nitrate and ammonia in the system. Another issue is phosphate. Phosphate is also another major element that trigger algal bloom if you don't take care of the phosphate. Again, it's the same. I'm going back to Norway because Norway is supposed to be a very clean country. And they have this problem of the lake with the algal bloom. And you can see here the water taken out of the lake showing big algal bloom because of high phosphorus in the system. So you can also remove phosphorus by designing phosphate anaerobically and also convert them back to aerobic. So operate the reactor in sequential fashion, and you can remove the phosphate. Phosphate is absorbed by the bacteria inside the cell, and you can modify the process depending on how much nitrogen, phosphorus, and carbon. Of course, most of the reactors are designed to remove carbon, but you also have to look into these two, nitrogen, phosphorus. So if you don't remove them, you're going to have eutrophication, where you're letting your wastewater. Okay. So let's look at the general process of the sewage treatment. So you need to remove, as I said, all the solids and carbon and nutrient like nitrogen, phosphorus, and then you need to, of course, eliminate pathogens. So you want to, the final effluent is going through disinfection process, either chlorinated, or you can use UV light or ozone. And recently, we have this problem of emerging contaminant, which is pharmaceutical product, antibiotics, nanoparticles. All these are going to, the wastewater system is not designed to take care of these. And these are now called emerging. Now people are trying to address how to remove these materials, because when they design this system, these are like in US, some of the systems are 50 years old, and they didn't envision that they're going to have this kind of problem. They want to just simply remove carbon, nitrogen, phosphorus. Now we have to deal with this new emerging contaminant. So again, going through this process for sewage, you go through the physical, biological, and chemical process. Physical is sedimentation, flotation, screening. Biological is where most of the bacteria come into play. You want to remove your carbon and most of the time. And then chemical process is to disinfect your wastewater. Screening could be simple screen, where the wastewater is, all the floating material can be, caught in this screen, and then you size reduction, you compress them and take it to landfill. And then sedimentation will remove the solids, that settlable solids. The membrane filtration comes later on in tertiary treatment process. And in the chemical process, you can do precipitation, adding lime, iron, and aluminum salt to form struvite. Or you can do adsorption and disinfection. So there are a lot of options for doing tertiary treatment in chemical, using chemical unit process. Adsorption, you can use ion exchange, and then disinfection using chlorination, UV light, ozone, a lot of things. So you need to make sure you have the regulatory standard of how much E. coli is our indicator organism that you're removing. So you have the chlorination, dechlorination, and now in US, if you live in marine system, you can do inject this into the down well below the salt line in the coastal system. So that's another way of removing these pathogens. So this is how the whole sewage treatment look like. You have primary, secondary, tertiary. So of course, all those physical, chemical, biological, chemical process, and then the solids are collected here, primary solids, secondary solids, come to this anaerobic digester. So this is where you operate anaerobically and remove the carbon, nitrogen in the sludge. And here you can convert them into methane in the anaerobic process, and you get some energy out of the system. After all this done, then you have the sludge drying system, and sludge is dried and taken out as biosolids. Okay. Just to show you, UV light is also used for tertiary treatment. These banks are UV light. So this how a unit operation look like: a primary, secondary, and tertiary when you design a system in sewage treatment plant. And this is the actual plant to show you all these units are in place. You have various operation of primary, second. These are all trickling filter with a filter that going through with the rotating arms. And you can make more sophisticated. Here you can have design of a treatment system to remove even the scum that is floating in the system. And then you can have the anaerobic digester and the dryer and the dry sludge going in. And then during this process, you get methane. So this you can make it as sophisticated as you want, as it's made as simple as you want when you design a system. And then now in state like California, they have water problems. So what they do, they treat the wastewater, and the wastewater they treated goes straight into the drinking water plant for treatment, and then the water goes to the drinking water. This is total recycle of water. All right. So what they do, after they go through primary, secondary, tertiary disinfection stage, it goes through the membrane filtration unit, which is tertiary treatment. And they use various size membrane from microfiltration to ultrafiltration, nanofiltration, and then reverse osmosis. And then the water comes out, supposed to be as clean. The wastewater is completely cleaned up. You can see what is being removed in each filtration, microfiltration, all the suspended and colloidal materials are removed, and ultrafiltration, you see the bacteria and virus and proteins are removed. And then nanofiltration, you see less molecular weight, organic molecules are removed. And the reverse osmosis will take care of all the ions. And then you get the pure water comes out. And this, of course, goes back into drinking water treatment plant, and it goes through another treatment process before it goes to public. So this is being used in places like California, where they have water crisis. And what is actually happening in membrane filtration is this operating under different pressure systems. Yeah, this is how the microfiltration work, ultrafiltration work, and you can see the pressure in the bar here. Just to show what is happening, how much is removed in different stages. You can see bulk of the solids and carbons are removed in secondary treatment process where biological activity. Okay, so a lot of carbon, nitrogen, phosphorus, and solids are removed in secondary treatment process. And then, of course, your E. coli should be reduced with the disinfection. Okay. So let's move on to how the sludge is treated, because how the sludge collected goes to anaerobic digester. It's basically another big reactor, and but you operate anaerobically. So so you have a complex organics that are converted into simple soluble organics, and then you have acidogenesis that convert the long chain organic to short organic chain compound like acetic acid, and then the last step, terminal oxidation step, where the methanogenic bacteria take these acids, convert them to methane and carbon dioxide. So you generally have 55 to 65% methane in anaerobic digester. Okay. This is just to show you this biochemical pathway of what's happening inside a digester. Carbohydrate, protein, fats are converted to simple monomer. And then you have acidogenesis, you have volatile fatty acids produced into acetic acid, hydrogen, CO2, and then you have this methanogenic bacteria take this acetic acid and hydrogen, CO2, convert to methane and CO2. Okay. And just to show you another way, pictorially, this is your anaerobic digester. What is going in, what gas is produced, and what is in the liquid that comes out. Most of the time, the concentration here is very, very low compared to what is going in, because the anaerobic digester is a well-matured technology, is being used everywhere nowadays. Okay. It could be a CSTR, completely mixed system, but anaerobically, big tank, depending on how much volume you're using. It could be a USB if it is a Aflo sludge blanket reactor. You can do CSTR in a big covered lagoon with a big plastic cover in a pond system, in a lagoon system. So in all the system, the same operation principle, you're converting the sludge, reducing the sludge volume, and you produce methane out of the system. And you can make it sometimes recirculate the sludge if you're losing it in the effluent. You can put a recirculating system, get the sludge back into the bioreactor. So when you're done with it, then you need to take the solids out, and the solids from the digesters are removed and dewatered and dried, and then they're disposed. Make sure there is not much heavy metals in them, and they can be combined with the soil amendment, and sell it as a commercial fertilizer, or sometimes use it as a fuel. So that's a whole operation of sewage treatment where wastewater is completely treated and solids are removed, and then the solids are dried out. Okay. So now, this is a very conventional process. Now what people are moving into is if you have enough land, or if you have land that you wanted to restore, they call it sewage farm. So the sewage goes through, of course, the basic activated sludge process, remove much of the COD, BOD, and then instead of going through the rest of the step, you go through the sewage farm, you release into a contained area and allow the plants to grow on it. Okay, so here is one in Detroit, Michigan, and people are doing this with a mild operation of sewage, and then most of them is released into the contained farm. Okay, so just to show you a picture of what they can grow, and they grow all kinds of native plants. And this removes most of the residual carbon, nitrogen that comes out, the plant takes up, and then the wastewater, after going through this farm, it goes into the tertiary treatment and released into the moving river system. And then this one in Berlin, you can see various schematics of where there are different discharge sewage coming in the farm. And then we have sludge drying areas, and then we have these dewatering trenches that come out. And then these are different treatments. Basically, different plants are growing. So this is very popular nowadays. If you have enough land, you don't have to put more sophisticated sewage treatment plant and make it a sewage farm. So these are different plants that usually grow in Western countries in this sewage farm. And it goes through trenches and released, and it's a highly engineered operation, but it is with nature. And you can see there's catchment treatment, bed treatment, slope treatment. Basically, you collect in the trenches, and then you have plant grow in the terrace, and then you percolate the water down and collect in the trench and release. And depending on different system, you have drain pipes. You can have a slope treatment following the gravity instead of pumping. So all kinds of things we can do. Another thing catching on in US is the wetland. So this is, I'm involved in locally, we call assimilation wetland. So when you discharge the treated wastewater, your regulatory standard is really a little loose, because the wetland is your final polishing step. So your COD, BOD in your wastewater could be high, and solids could be high, but the wetland will take care of your final process. And in this way, what they are doing in here is, what happened is, because the cities are growing, more housing coming up. So the natural wetlands are built around with a lot of new houses. So they lost the natural stream of water into the wetland. So they are dying out because there is no water. So they're restoring this wetland by pumping this partially treated wastewater into the wetland. And a lot of this wetland being improved. And we have a couple in where I live. I got involved. So here is, I had a list of the sewage. Tubular is one of them where I live, but I don't know why it didn't show up. So this is, we call that, using wetland treatment system, we call it assimilation wetland, because these are wetland already there, they're dying out, but we are restoring them. That's why it's called assimilation wetland system. Okay. Now there's also constructed wetland is becoming very, very popular in European countries. And you basically construct this wetland in different cells, and the wastewater comes in, doesn't even go through sewage treatment. It goes straight into this constructed wetland. This is where most of the treatment takes place. And it goes through, of course, it's engineered highly. And a lot of plants grow in there, and it just percolates down, and the waste is collected after it goes through. And you can see the difference in BOD sewage that comes in here, solids that comes in there, and what is coming out, and in the effluent coming out of the system. You can construct them in vertical constructed wetland or horizontal constructed wetland. So different designs out there. So this is another way of doing it. So here's another system to show surface flow constructed wetland. And this is just a picture, and this is the actual wetland in Spain, Valencia, and before the wastewater led into the ocean. So they don't actually have a sewage treatment plant in this particular town. All the wastewater going through this wetland, but highly engineered process. Okay. So you collect the wastewater and basically remove the pre-treatment, use the pre-treatment to remove the solids, and then goes through septic tank, and then goes through secondary treatment is our wetland. Instead of activated sludge, instead of a trickling filter, you have this wetland as your treatment system. So here's the actual picture of a constructed wetland in Spain. Okay, so this is working really good, and you can see the treated water coming here before it is being discharged. So what's happening here is most of the carbon is degraded both aerobically and anaerobically, depending on the depth of the wetland. Organic matter is also removed by a lot of different reaction takes place in the wetland, because there is a lot of different group of bacteria there. In the last class, I talked about the redox potential of the waste. So you have sulfur-reducing bacteria, nitrate-reducing bacteria, and aerobic bacteria on the surface. So this is how the constructed wetland look like. And just to show you some data, what activity is going on in this process. So it's a little bit engineered process, but we are trying to put nature do the bulk of the removal of this carbon. So just to show another profile of vertical constructed wetland, then just showing you what is being removed, and the BOD nitrogen and solid removal in this wetland system. And this is a horizontal flow wetland. So you can see a little bit longer the system is. Okay. If you look at it, it has influent, and it has this filtration permeable line. It has an impermeable liner, so there is nothing seeping into the groundwater. And then it percolates down, and your plant's going to take care of nutrient uptake, and then the effluent comes out, and treated waste come in, collected in a pond before it is being discharged. And then this wetland also produces sludge. So how do you take care of the sludge? Sludge is treated in, again, a wetland. Okay, so here is one in Denmark, and they pump the real sludge into this wetland and this direction, and the plants will take care of the nutrient and uptake this nutrient, and it percolates down the sludge, and then it collects in a drainage pipe, and then it's being the water that can come out can be pumped out. The sludge is retained in this wetland. All right. Just to show you actual wetland, here is the wetland for the sludge treatment in Denmark. the waste coming in a storage tank and put in different cell. They go through in cycle, go through first cell, second cell, third cell, and come back to first cell. So you don't want to pump the sludge all in one big area. And alternatively, they pump into different cells. And this is how it looks like when the sludge comes into the wetland. All right. And then after running, few cycles, so you can see the sludge is kind of dry, and then nutrient uptake, the plant started to grow on this. And this is the actual micropollutant removing wetland system. I talked about antibiotic and personal care product. So here is an engineering system they built in Spain, and this is the actual site. You can see this wetland right there in the town. And this is how the design looked like. They have the MHOP tank, vertical flow, constructed wetland, horizontal flow, and free water surface wetland. And these are the design of how much wetland in that area and the picture I showed you. And this is specifically designed to remove those emerging contaminants I just talked about, all right? The antibiotic, personal care product, nanomaterials. And this works really well. They've been operating for a few years now, more than 3 years, and it takes care of all those pollutants that normal sewage plant cannot take. And there is a list of chemicals that this constructed wetland can remove. A lot of antibiotic included, the antimicrobial compound triclosan, then common over-the-counter pharmaceutical compound, those can be removed, but in the conventional plant, it's not. So this is where the Western countries are geared towards now. Sewage plant now, they're combining with wetlands treatment to take care of the domestic. And then we have a lot of rural population, and everybody has to have septic tank or individual sewage plant. And this is highly regulated. People come and inspect your home. So you need to make sure you're doing it really good, and you have to put chlorine tablet every week. So make sure your pathogens not getting out. So this is for system that is in rural area. So you have your rural area that your waste collected, and then if you have, of course, a farm or you have enough area, you can have this absorption field after you go through a conventional septic tank system, and you can spread it out in your field, and the waste can dry out and can be taken, treated water can go in later. So all the system, again, basically concept is microbes and oxygen, the limiting if it is aerobic conditions. So you kind of encourage the growth of bacteria in the system and other microbes as well. So you can create a large lagoon, holding tank, and provide oxygen and sunlight, and bacteria will take care of your excess carbon. Okay. So when you do this in a big lagoon, there is a lot of activity going on. They have this aerobic organism here, facultative bacteria here, and then anaerobic zone at the bottom. And then you don't have to pump oxygen. Most of the oxygen comes from your natural system of wind and the surface. And then you allow the algae to grow. The algae during photosynthesis is going to produce oxygen as a natural system. And then the anaerobic process will take care of what is being settled down. So this is kind of combining natural carbon cycle in a big lagoon system. So this is kind of some cities are testing this out in US. Instead of building really highly engineered process, they kind of simplify this in one big lagoon. So you need to make sure you design, again, depending on how much wastewater coming through, you need to design the lagoon accordingly, and also the carbon loading, nitrogen loading. So a lot of some design concept go in there when you design your pond system. So this, I just went through some basic concept and little bit of commercial reactor, and then I talked about what is being popular nowadays. Is constructed wetland, assimilation wetland, and sewage farms are getting popular now. So now I'm going to specifically talk about one case study which I was involved. I'm going to talk about how we removed and design a system that is highly contain high concentration of nitrogen. Carbon is not a problem. In this case, nitrogen is a problem. So here is a shrimp aquaculture system where they feed shrimp 40% of protein in the diet. So the shrimp in aquaculture system that highly concentrated protein in the diet. So most of those diet they feed sometimes is not being eaten by the shrimp. So the protein goes into the water and then converts into ammonia, right? So the ammonia is what we are trying to remove, the nitrogen in the wastewater. So I was personally involved. I'm going to, show you how we designed the system and how we implemented in large scale. Started out with small 2 liter in lab and built up in a big system, an actual commercial system, to walk you through the whole process, how we did it. So a little bit of introduction about how we got involved. Because of aquaculture is getting popular, most of the seafood is now coming from aquaculture, not natural system anymore, not wild caught fish. Most of the fish are grown in a pond system, okay? Most of them are coming from Asian countries, and China is really big, and other Asian countries do a lot of aquaculture, but the Western countries' contribution is very less. And the value-wise, in terms of dollar, again, China dominating in aquaculture market, and Asian countries are excluding China. Again, I told you the 72% of all seafood is aquaculture. Only 28% is wild caught. Within this aquaculture system, we have mostly freshwater fish aquaculture, like here a tilapia farm is one common one. But the one that we have worked with is this crustacean farm, the shrimp farm. Okay, shrimp farm is 9.5%, but shrimp is, in terms of dollar value, is high. $22.7 billion compared to fish 40, even though this is 50% of all the aquaculture, because shrimp is price-wise high. So shrimp is getting very popular worldwide. And let's again look at how the shrimp aquaculture look like. Again, China dominating, all right. Indonesia worldwide 10%, and the US aquaculture shrimp is very, very less because of the high regulation here. They couldn't compete with the import. Some of these countries have lax regulation. Here we have high environmental regulation. So one of the regulation is you cannot have shrimp aquaculture close to the ocean. It has to be 75-mile buffer from your pond system or into the ocean. They don't want any disease getting into ocean and wipe out your natural fish. So we have, that's a major problem in US. So that's why this is very, very low in US. Within the shrimp, the one particular species is produced worldwide, 57%, which is Lithopaneus vaname. And then the tiger prawn is another one, like Paneus monodon, so it's second best, okay. This one is very high in terms of production, mainly because you can grow them in high density, and also you have some disease-resistant variety you can produce nowadays, disease-tolerant species. If you look at the US market, the production went down because, again, they couldn't compete with cheap import. So most of the farmers just gave up. They lost money in the process. So the government got involved mainly because of we are importing a lot of this shrimp, $3.1 billion trade deficit from other countries to meet the demand in US. So the US government said, gave up some research funds and looked at universities, said you need to help the farmers and try to, increase the production. So we got involved. This is called US Marine Shrimp Farming Program from USDA. And these are the institutions got involved. We are involved in taking care of wastewater. The other guys are involved in different research like genetics, disease and all the other cases. Okay, so the main objective of this project is to increase the domestic shrimp production and make it competitive. Okay, so again, the restriction in the US, in the other countries, the pond culture, they close to the ocean, they can easily pump the seawater from ocean into the pond. They don't worry about pathogen introduction into the ocean because of lax environmental regulation. And they use antibiotics. In the US, you cannot use antibiotics. And then all these waste chemicals going right into the ocean, and then they have low feed conversion ratio. But in the US, we have the restriction. So we need to make sure that it is biosecure, away from the coast. So we come up with this design. Most people now use this called recirculating raceway system, where there is no water exchange. Everything is, once you truck the seawater from the ocean, you don't go back again, because that is a major expenditure. So we wanted to treat the wastewater and recycle that seawater over and over again. And so nothing is wasted, everything is recycled. So it's biosecure, and in a controlled environment, we try to have a high conversion ratio, feed conversion ratio. As a result, when we do that, we produce this concentrated effluent high in nitrogen. It looks like this. Very, very sludgy and high protein diet cause high ammonia in this wastewater. When you have high ammonia, it's going to kill the shrimp. So it's kind of a challenge for us to design a system to remove this nitrogen and try to extract as much salt water back into the system and dry this sludge. And you need to repurpose this sludge. We need to use this sludge by growing salt-tolerant plants. So the key point here is you need to make sure that your shrimp, the ammonia level should be below 3.5, nitrate should be below 15.2, and your nitrate is 150 milligrams per liter. So you want to make sure to bring these concentration less so the shrimp will survive, so you can recycle the water. So we just looked at using the nitrogen cycle. So we know we started with high in ammonia. So we wanted to use this a nitrification process, convert this ammonia to nitrate, allow the bacteria to grow, the nitrosomonas, the nitrobacter to do this oxidation, and then do the denitrification step, convert nitrate back to nitrogen gas. So you remove this nitrogen, this cycle, partial cycle of nitrification and denitrification in our design. So we obviously look at sequencing batch reactor, and then for denitrification step, you need to add carbon to the system. So we use molasses as one of the cheap carbon source. Okay. So this is how a sequencing batch reactor look like. You operate one tank sequentially. So you fill your reactor with wastewater, do aerobic process to do your first reaction, which is nitrification reaction, conversion of ammonia to nitrate. And then you switch the reactor to anaerobic mode. So the denitrification takes place. The bacteria in the system can convert nitrate back to nitrogen gas and do one more polishing step to remove any carbon left over. The aerobic process will take that carbon quickly. And then you settle and decant, and the water goes back to recycling and wait for another batch to come in. So that's our simple Sbr system we designed. And then the sludge that comes out, we have to repurpose the sludge. So we grow this sludge because it's a high salt content. We use to grow the marsh plant. Okay, so we kind of sustainable way. So everything is used up, nothing went to waste. Okay. So that's how we started the whole concept of using sequencing batch reactor following the nitrogen cycle principle. Okay. So again, this is what's going on biochemically. You operate aerobically to drive the ammonia down to nitrate, and then you switch to anaerobic mode, then nitrate goes back to nitrogen gas. So you have same one reactor operated in two different cycles, aerobic cycle and anaerobic cycle. We started out with a small lab scale reactor, which is a very, very simple plastic bucket covered, and then you have a mixing device, aeration system for aerobic process. We monitor different parameters in the wastewater, and we collected one from recirculation raceway, another from pond system that has the bead filter. The sludge is collected here. And then we did the analysis. If you look at it, these are the things that we are worried about: ammonia, nitrite, nitrate. We want to bring this down to 3.5 and all this to the standard that we set. And then the carbon is not a problem if this 1500 can be removed easily by any aerobic process. So we kind of design lab reactor operated. We want to make sure how many days we have to operate aerobically and anaerobically. So we kind of played around, and just to show you a simple system, and you can show the total COD removal by operating aerobically and anaerobically in different hours. And this is soluble COD removal in 4 different reactors. And then you can, this is where you need to play around with your aerobic and anaerobic sequencing. So when you do aerobic operation, you can see the ammonia goes down, because that's where the ammonia is converted to nitrate, right? And so you need to see how many hours you need to operate aerobically and then switch back to anaerobically. So this is how we played around with the sequence. And you can see when you switch to anaerobic mode, the nitrite went up, and then nitrite came down because nitrite is converted to nitrate. So the system is working, just we are playing around. And then you can see the nitrate in the system, NO3. So NO3 comes up after the nitrate went down, and then anaerobic mode, the nitrate goes down. And then at the end of the process, it becomes carbon limiting during denitrification. So the system nitrate build up. So we need to add a little bit of carbon to grow the bacteria to make, because the excess nitrogen, you need to have carbon to balance this out. So when you add a little bit of carbon in the form of molasses, all the nitrate went down. So this is our initial lab scale reactor to figure out the sequencing step. And you can see during the process, we removed 20% of the sludge, and the sludge removed because of the bacterial action. And then we played around with the optimum temperature and salinity, because this operation of these reactors is need to optimize for local conditions, because some farmers use low salinity to grow shrimp as 5 parts per thousand. Some farmers grow the 28 parts per thousand at 30 parts per thousand. And then we have temperature. Some shrimp farms are in the southern part, which is high temperature. Some of them are in northern part. So we want to make sure our SBR work in different conditions. So again, we went through optimizing the same system in a small scale reactor. It's only 500 ml reactor in a 1 liter flask, small reactor. But we operated in different temperature. We kept the salinity constant and did the 3 reactor for treatment. This is our sequence based on our previous study: 2 days aerobic, 3 days anaerobic, and 2 days aerobic, 1 day to settle. So total operation of 7 days for 1 cycle. And just to show a different temperature when you operate the reactor, most of the temperature worked well. This is the aerobic, anaerobic, aerobic settle sequence. And the high temperature, 45 degree, didn't work. It removed, but it didn't go all the way down, ammonia concentration, the way we wanted. But 45 degrees is extreme, but we just put in there for global warming, so it could reach 45 degrees some days. So. Then we have the nitrite, this temperature, same case. When it is high temperature, the nitrate removal was not good, but all other temperature was okay. The nitrate is the same case. So because the bacteria is not adapted to this temperature, the bacteria come from 30 degrees centigrade when the system that is operated, the sludge. And then the carbon removal also showed the same process, total carbon removal. That high temperature did not work. And then we kept the temperature constant to 22 degrees, and then we operated the sludge in different salt, because some farmers use low salinity, some farmers use high salinity. You want to see whether is there a problem operating our Sbr with different salinity. Our temperature is the same, but the salinity level is different. But again, most of the salinity work except the low salinity did not work, because our sludge originally came from 28 parts per thousand operated farm. So depending on your condition, your bacteria system not adapted to this in this lab setting. Okay. And then nitrate level, same thing. Your 5 parts per thousand did not work, but 28 and 40 really worked well. And this is your NO3, same story. 5 parts per thousand did not work. And this is total COD, okay? So based on this, our 45 degree is going to be a problem, and 5 parts per thousand is going to be a problem, but except other conditions, our system is going to work really well in the lab study. Okay. And then the sludge also reduced. You can see the 21% reduction of sludge in good treatment, and 45 degrees temperature sludge was not reduced as much. And the salinity is the same thing. So the good operation of 28, 40 parts per thousand, we have good sludge reduction compared to the 5 parts per thousand, less sludge reduction. So there is this condition, you need to make sure that you, depending on where you take your bacteria, you need to optimize the condition for that particular system. So with this lab study, then we come up with this plan, how you're going to do. You have this recirculation system, and then you can get the sludge and put in a holding tank and design an Sbr, sequencing bed reactor. After this removal of the ammonia nitrate, and the water goes back, and the sludge can be reused, okay? So you don't have to truck your salt water from the sea every time you grow. So you just kind of one time bring the water, and then you recycle the water back over and over. So this is our actual farm, and you can see this closed system of recirculating raceway. This is 228,000 liter. This is where a 2,000 shrimp per square meters, high density shrimp. And this is where your ammonia builds up, nitrate builds up. So we need to remove this sludge, okay? So my grad student went there and set up this Sbr, and you can see we had a control. Is just without sequence, just to kept it anaerobically. Then we have the Sbr that is operated with this 2 days aerobic and anaerobic sequence. This is how it looks like. This is your control. This is your Sbr. You can see that after 1 week of operation, look at the water, the solid sludge here and sludge here, okay? So just to show you what this is the big system that my grad student operated. This is total ammonia nitrogen, started out high and went down from 2 days of aerobic. It kept it to the condition we wanted, less than 3 ppm, okay? If you look at nitrite, aerobic, anoxic aerobic system, nitrate went up and then nitrate came down. So it worked perfectly as we designed in the lab. And then the nitrate concentration also went up, and then it came down in this large scale operation. And then if you put all the nitrogen together, add the number, it shows the sequence of going up, coming down, aerobic and anaerobic operation, okay? And then we wanted to make sure this works in a different farm. So that one was in South Carolina, and this one was set up in Mississippi. So here they had the smaller system, and this is again a raceway of growing shrimp. So they have this waste coming from this holding tank, and then collected and converted into this Sbr. There's a simple operation. It's a big plastic bucket, and you have this mixing device to make it aerobic and anaerobic more, okay? We had a control, and we also had Sbr system. So this is because of the building has two systems. So we built two different areas, this Sbr system, greenhouse 1 and greenhouse 2. And here we wanted to make sure that we can save some money by doing partial nitrogen cycle. So when you do your nitrogen to ammonia, ammonia oxidation, ammonia to nitrate, you go through this step of nitrite and nitrate. So we thought, why not we stop the reaction here, and then we switch the reactor to anaerobic mode. So because when you do your denitrification, it goes the same step, nitrate to nitrite, and then nitrogen gas. So we can save some oxygen, save some carbon if you do partial nitrification, denitrification steps. So we kind of played around with this nitrogen cycle. So we kind of going through all the way and coming down. When we reach the peak of nitrate, we switch the reactor to anaerobic mode, and then go straight to nitrogen gas. Okay. So this is just to show you the data. We have a control. We have the greenhouse 1. You can see this nitrite data during aerobic anaerobic sequence. It just came down to the way we wanted. This is nitrate, NO3, built up and came down, okay? And this is ammonia nitrogen. So when it came down during aerobic process, all the ammonia came down. Okay, this is control when there is no SBR. This is nitrite, and this is a greenhouse 2, okay? So this system worked as we designed. We played around with the partial nitrification, denitrification step. And then we want to know what kind of microorganisms is in the system. So we looked at them, molecular analysis, and most of them are this Paracoccus and Pseudomonas, and in the water that doing this steps of denitrification and nitrification. And want to show what a gene is turned on, and during denitrification step, this NOS gene turned on that does the nitrous oxide reductase. And a lot of those time when we take sample, these are the genes that we found expressed in the wastewater. And then we had this solid built up, the sludge. So the sludge has to be repurposed and used some way. Because of the high salt in the sludge, you cannot use them for normal crop. So we wanted to grow the salt-tolerant grass called Spartina. They use this Spartina to restore the coastal land loss. So they go plant this grass back in coastal system. So we kind of idea is to sell them to the salt nursery system where they can use this sludge to grow this plant. So just to show that this plant, Spartina ultriflora, can grow in this biosolid come from greenhouse after went through the SBR process. We grow this plant, and you can show the control with normal without sludge, but it received normal nitrogen, phosphorus. It grows better than what is normally grown in the greenhouse. Okay. And just to show this plant average growth was better in the biosolids come from SBR. So we wanted to make sure that we met the design criteria to make that our ammonia is low, our nitrate is low, or nitrate is low, so the shrimp can grow. So we don't have to go all the way to 3.5 because we can dilute the water if you need it to. So when we dilute it, it's 0.33 milligram per liter ammonia that you meet the criteria. But we went through completely 3.5 and below. But just to show you, you can sometimes live with partial removal. Okay. And then we did the cost benefit analysis. We put all this, how much it's going to cost for a small farmer. So we looked at the how much to truck your seawater with the 20 parts per thousand salinity, and then how much to dispose the waste, and how much the biosolid going to cost, and how much you spend on setting up your SBR, and you need a pump to, pump the system, how much molasses you're using. If you look at the price and the savings and the cost, you could save in your first cycle. This is a small operation just to show you, for a 200,000 liter recirculation, the sludge can be treated and the wastewater can be recycled. And in your first cycle, you actually save. If you don't put the plan, you have to spend this money to truck your water, to dispose the solids. So you could actually save within your first, get your money back when your first time you do your system. So the cost benefit analysis showed it works. So this is one of the successful SBR operation that farmers are adopting nowadays. Okay, so we just to show you how we built up from a small 0.5 liter to a bigger, 200,000 liter aquaculture system where your nitrogen can be removed and farmers can reuse the wastewater using our design. Okay. A lot of people funded our project, and just to acknowledge some of them. And we, of course, not everything went smooth. We encountered some problem. You can see when you do aeration, because of high protein in the waste, we have this foaming problem. So we have to have, foam control sometimes. That's a foam spray to control your foam. Okay. So we had some problem, but we addressed it at the same time. Okay. I will stop here, and if you have any question, I can answer.
Okay, thank you so much, Prof. Raj.
All right.
You can display the question from the audience.
Okay.
Okay, Prof. Raj, first question from Bu Reni. Currently, there are two issues on municipal wastewater especially during this pandemic.
Yeah.
The presence of the SARS-CoV-2 in the wastewater, and also the excess of chlorine due to the extra protection, suggesting because this is one of the method that suggested to kill the potential virus. Maybe you have any other suggestion instead of using extra chlorine, because it has some side effect. Right?
Yeah, currently that's what they're doing here. Wherever they're using chlorination, they're using excess chlorine to kill this virus. But some cases they're using UV light and ozone treatment. That's also working for us. And in plant where I live, they put extra UV light. So they have chlorination and they have additional unit of UV light. So depending on, how much virus is in the system. So yeah, that's a good question. So that's a new problem to address. So yeah. So ozone, UV light seems to work really well, take care of those RNA degeneration. So yeah.
But Bu Reni, do you have a follow-up question regarding this question? I mean, if we use UV light in the in the municipal wastewater, maybe you have some example of or study cases that how we can apply UV light in wastewater system.
Yeah, I mean, you can design a system where when the effluent comes out of your system, you put banks of UV light, so it passes through, and you just hold it for a few minutes, and it goes through a series of banks, like not one light. There is a series of light. The water goes through and comes out, and you have a lot of lights. And by the time it comes out of this, we call that UV light bank system. Most of the viruses RNA is degenerated. So
it should be effective for.
Yeah, it's like a retention time. You're keeping the wastewater in a UV exposed system. I would say like a minute, maybe. I'm not sure exactly how the system is set up, but I went and looked at it. It has a series of banks of UV light. It just goes and comes, and all these places the UV light. So that seems to work. Yeah.
Okay. Thank you, Professor. Now, second question. Still from Bureni, we currently try to increase the biodegradability of white rot fungi using ozone treatment prior to the bioprocess. Maybe do you have any idea? Do you think the presence of ozone will have any effect of the bioprocess performance?
Increase. So the ozone treatment, you're using it for the raw material. Where you're using the ozone?
I think so, Raj. Rainy doing my student and I using for the textile for the color. So
Okay.
the color for the batik wastewater.
Okay.
And then, is there any possibility if we use the ozone first for the dyes before treating with the fungi? Is the ozone will have some affected to the fungi or not?
Not to the fungi. No. But once it comes out, you don't have any residual ozone in your waste. I think its fungi should be okay. Because we did similar system, but we didn't use the ozone. We used the what we call sonication process. So the sonication process kind of softened up this dye, and so it kind of helped a little bit to improve the performance. So yeah. So you're using same pre-treatment instead of, I mean, ozone should not have any problem with fungi. These white gut fungi have really robust system. Yeah.
Okay,
But I didn't try ozone, but I think it will work. Yeah. So is there a high concentration? What dose you're using? You're playing around with it right now?
Yeah, we are still playing around, so we are not yet fixed. But Bureni, it might be your.
It's between 3 to 5 ppm.
That can, and 3 to 5 is low enough.
It's low.
Yeah. The fungi will tolerate that. Yeah. After you treat them, the residual ozone will still there. You hold it and treat them, and then you're going to expose them to fungi, right? Yeah.
That's right. Yeah.
Yeah, there's going to be some ozone going to be lost, and then there'll be some residual ozone left. And when you started with 3 to 5 ppm, you're going to end up maybe 50%, 60% still there. And I think the fungi will be okay at that low concentration. Yeah. That's my opinion. So it's a guess for me because I didn't work with ozone before. Yeah.
Okay. Thank you, Raj.
Thank you, Prof. Raj. Okay, the next questions. Do you have any? Can you give some guidance about how to properly design the disinfection process, especially for dosing and determine the retention time? To make sure that the microbial parameters will meet the effluent standard, but the residual chlorine is still at a safe level in the effluent.
So this is for what municipal wastewater? Any particular wastewater? It depends on where you're letting the wastewater out. The chlorine, if you're letting it out in a wetland or letting it in the river, less than 0.5 ppm should be okay. The residual chlorine shouldn't be a problem. So, but if you wanted to do what dose you need to apply to kill effectively most of the microbes, you should have to do a simple design in the lab just to work small scale, figure it out how much chlorine you need to eliminate. So what's your removal efficiency? 95% removal, 100% removal. So you need to play around with the chlorine dosage. But as the letting it out into the river or stream, 0.5 ppm residual chlorine seems to be okay. Yeah.
So around 0.5 ppm.
Yeah. Whatever left after the treatment, you don't have to worry about. That's going to affect the natural system. Yeah.
And how about the detention time? Is there any best practice for for chlorine?
Chlorine?
Yes.
For disinfection in wastewater system, they hold it in a tank, and it's just an hour basically in large scale. Yeah.
Around 1 hour.
Yeah. Because of the amount of wastewater going through, you can't hold it long. So a big volume of wastewater coming through. Yeah.
Okay, thank you, Prof. Do we have next question? Okay, this one is from the participant, Awalina. Do you have any suggestion to prevent emerging disease on shrimp culture without implementation of antibiotics?
In US, what they use is, if it is a bacterial disease, we are using bacteriophage therapy. So we have most of the Vibrio. Vibrio is one of the bacteria that cause disease in shrimp. So we have a Vibrio-specific viruses they introduce to control Vibrio. So that seems to work because we cannot use antibiotic in US. So that's the only way to control. So far that seems to work. Yeah. Of course, viral disease antibiotic is not going to work on viruses. So sometimes we have this viral disease that's going to wipe out. And we have experienced a lot of farmers going out of business with viral infection of shrimp. But bacterial disease we can control without antibiotics. Yes. Yeah.
And how about the viral disease, sir? Normally, how they handle the viral disease?
The viral disease, once it gets into the system, they try to harvest the shrimp as quickly as possible to try to save the crop. And they have to completely let the water out and bring in fresh water and treat the water with UV light. It's a big expenditure for the farmers. Yeah. We had a couple of farmers went out of business. They had the viral disease. Yeah. Okay. Infection. Yeah. It's called white spot. It's one of the common one. In this shrimp. Yeah.
It comes from the air or from the.
I think it's introduced from within the shrimp. The shrimp itself is not totally disease-free. It just when they come from the nursery, they buy the shrimp from in a small larval stage. It comes with larva. So the supplier not doing a good quality control. So they're selling into the farmers. Some of them has the virus in them. So yeah. That's how it gets into it. Because most of this farm I showed you is all biosecure. It's completely contained. So there's no other way virus can enter. Yeah. So most of the time it comes with where they're buying the shrimp to start with. Yeah.
Okay, thank you. Next question is from Rizal Malik about the selection of the reactor types that you have mentioned before. I've read that USB are preferable for treating high organic content due to high yield of methane that produce. I want to ask, is it efficient if I use the USB for treating low organic wastewater but containing toxic substance such as recalcitrants or POPs?
Yes. You have to have this carbon nitrogen ratio. If your aim is to remove POPs in your wastewater and any other toxic substance, but it starts with low COD, you need to provide some carbon to keep your bacterial number high. So for example, when my denitrification step, I ran out of carbon, so I had to add molasses to the system. So you had to watch your carbon nitrogen ratio in your system. So in wastewater, generally 30:1 ratio, that's what they follow. Okay, sometimes 20:1, so depending on wastewater. So for every 30 parts of carbon, you need 1 part of nitrogen. So and then if you have more toxic substances, if you have less carbon, you got to add more carbon to the system. Yeah, you got to have a balance of having your biomass number high. Yeah. But USB will work for sure. There are a lot of research done on USB in using personal care product and some industrial wastewater that has some toxic component to it, especially pharmaceutical wastewater. Yes.
Maybe some addition question. Beside the C/N ratio for the toxic substance in wastewater, what kind of other parameters that we need to pay attention, Prof?
If the wastewater is a lot of carbon in the system, most of the time it's carbon, nitrogen, and carbon phosphorus ratio you need to monitor. In rare cases, you need to add some trace elements. Sometimes they lack iron because they need a little bit of those trace elements to have proper enzymatic activity in bacteria. So people add some iron to supplement these trace element requirements. So most of the time it's carbon, nitrogen, carbon phosphorus ratio. Okay, so you need to have 100 for every 100 parts carbon. You need 1 part phosphorus. So it's 100 to 1 for phosphorus, 30 to 1 for nitrogen. So you need less phosphorus than nitrogen. Yeah.
Okay, thank you. Next question is about the potential of waste-derived materials industry in the future. Do you have any opinion about this one?
What the potential of waste-derived materials? What do you mean by waste-derived materials? You mean the waste itself? Waste-derived materials industry. I'm trying to catch up that word. What bit? I'm not quite getting that. Tjandra, can you help me out? What he's trying to say? Waste-derived material?
Waste-derived material
industry.
Is this the Freddy? Because you are in the Zoom, probably you can explain more.
Is this the like recycling, like electronic?
I think so. Yeah, I think
like
so.
Electronic chip board and all that thing, circuit board, all that stuff. Is that what you're trying to do? What,
yeah, I think so. I think so. It might be. I would like to try to get the. It's mean that to use the waste for the resources.
Okay.
I think so. So that one is a waste derived. I think so. That one is a. It might be relating with the circular economy, something like that, George.
Yeah. I mean, for wastewater, today I'm only talked about wastewater issues. So most of the time, wastewater is coming from production systems. So, but if you're dealing with, for example, you trying to recover material out of the waste and then you derive wastewater, you can design a system for that. But if it is a solid waste, that's a different research. But so if you're, for example, washing your electronic circuit board and you're going to generate the wastewater, and then the circuit board may have some component that you want to treat, yes, you can design based on what chemical substances in the wastewater, you can design a system. Yeah. So you need to characterize the wastewater first and look at what kind of waste is in the water. So yeah.
So based on the quality or the properties of the component inside the
that's right. That's right. What kind of chemical is in that water? Yes, you can design the system. Yeah. I mean, most of the Asian countries, they now deal with this old computer parts and stuff, and they try to get stuff out, and then they wash the boards and they generate wastewater. Especially in India, they are doing it, and they have some problems. Yeah, they have. Most of the time, it's not much carbon, but there's actual chemical in the circuit board itself. That's what they're trying to deal with. So in that case, you add carbon to grow bacteria, because the chemical itself is not going to act as a growth substrate for microorganisms. So you need to add some simple carbon and then expose the bacteria to this, whatever chemical is in that waste, and hopefully they will remove that. Yeah.
Okay, Prof. Thank you so much for the answer. Do you have any other question? Okay. I think we have one question in the chat from Awalina. Due to the recirculated aquaculture system, is there a certain critical time to re-add some nutrients in the system? Or maybe do you have any best practice experience with this kind of problems?
The Sbr, that's why you need to design how long you need to do your operation mode, aerobic, anaerobic sequence. Okay. In the aquaculture system itself, where you produce your shrimp, that one is already optimized by the farmers, like how much oxygen goes in the system, how much feed goes into the system. But they're trying not to waste that much nutrient, but they ended up, because of the amount of protein in the diet is so high, so they couldn't control feeding them. They tried to feed in a timer every few hours. They put the pellet. This is the feed that goes into this production of shrimp. So they set up the timer. It comes the conveyor belt. Every few hours, the conveyor belt moves. It puts the pellet into the pond and where the shrimp eat. But even with that, they produce a lot of nitrogen. So they're trying to figure out optimum way, but anything they try to optimize, they still have more nitrogen because of the high protein. It's gonna, as soon as it hits the water, it's going to dissolve, right? Some of those protein going to dissolve in water, and then the bacteria in the water going to convert the protein to ammonia. So that's what we are dealing with in this particular system. Yeah. They try to do a lot of different ways to optimize the whole aquaculture recirculation system, but they always produce high nitrogen in the wastewater.
Okay, sir. Okay. Any other question from the participant, or Bu Reni, Pak Tjandra, maybe do you have any question to Prof Raj? We still
okay, for things for me is I think it's already late also for Raj.
If you have any, yeah, no problem. I just check in the clock. You have 7 to 9 class, right?
Yeah.
I'm fine. Yeah, I'm at home, so yeah.
Okay, since there is no further questions, I would like to thanks to Prof Raj that's already given us a very good presentation and insight into the treatment.
Things are the
It's not from my home. The dog is not from here.
Yeah, I think the next class will be the next 2 weeks, right?
That's right. Not next week, the following week. Yeah.
Yeah, the following week.
Yeah, I'm gonna talk about biofuel, basically lignocellulosic ethanol production and microbial lipids. I'm gonna combine together. Okay. The next topic. So ethanol production, microlipid for biofuels.
Yeah. Next week, Asok Pandey will give a lecture.
Oh, really? Okay.
Yeah, on Wednesday, not on Friday.
Okay. Good.
Okay.
That's good. You're bringing in some other expertise. That's nice. I'll see you guys in 2 weeks from today. Yeah. 2 weeks from.
Okay. Thank you.
All right. I'll say good night now.
Thank you, Pat.
Thank you. Bye. Bye. Good night.
Bye.
Thank you.
Bye-bye.
Prof.
Thank you.
Bye.